Thank you for the invitation and the possibility to be here at the conference. It was Marcello who asked me if I could not give a presentation on my paper, the Modularity Codes. And the paper is really a bit complex or goes very much into the detail. And today I want to give more visual approach how to relate codes to modularity. I have the presentation and several other presentations on a small USB card. It's like this, and you can flip it, and everyone can take one. There's also a CV of mine included, and when you go to the last page of my CV, there is a list of relevant papers with hyperlinks you can click. Come to the papers. If you cannot download it from the publisher, you may just find it somewhere else, and if you don't find it, then you can contact me. But you don't have to take notes or anything, and you may use some of the material for whatever you want. If you cite me, that's okay. So, a short word. Rechenkraft is a German public club. It's a non-profit organization that is engaged in scientific projects that are based on distributed computing. And hopefully, with this computer infrastructure, we will be able to do some data analysis in the future when the list of codes is so long that we cannot handle it manually anymore. And what I want to do with this visual approach is to show you how one can think of modules through codes. So in biology, modules are defined as functionalities realized through multiple components in a very short definition. But actually, this is what agreed on is this function aspect of a module. There are different definitions, but function is the unifying theme in modularity in biology, at least. So, but if we have a code that is composed of an adapter and two components that are mapped by this adapter, then we can have this tripartite structure. And if we just stretch it, rearrange it to have it in a row, and then we can align such codes. And when we do this, we can play this, what I call molecular Tetris. Yeah, we can check all the codes that are in a cell or in a system and see how they align. Yeah, and we come up with something like this. These are I did not number it so that they are not different. In my paper, I used for A1, 2, 3, 4, and so on, and different components so that it's clear this is not the same code, but it should symbolize different codes. But we may then find at the same time the expression of or the presence of active components that may form a line, and maybe we have then separate areas or functionalities within a cell that are can be unified or divided, however you would read this. So we end up with sets of codes that define modules. And this is the main pattern I want to point towards. I have not yet done any empirical study, but this is one of my plans to have a follow-up paper on the modularity codes where I go into the databases and analyze described modules in biology and look if among the components of those modules, what are the codes? Are there codes in these modules? So let's say every C is a component. When we have a metabolic pathway, for example, in microorganisms that use different carbon sources for their living, then there are different enzymes and there are different codes. And so based on these functionalities, I want to check the number of codes per pathway or per module. And if these components have more connections among each other than to completely different components in the rest of the cell. So metabolism is only one part of a cell, yeah? And there must be interactions with the rest of the cell. But if the density of codes or components mostly pertain to a specific module, then the interactions with the rest of the cell are fewer. And so this says us something about how this is organized, and for the first time that modularity could be explained by means of codes. This is the basic idea how we can explain one of the fundamental principles, that is modularity, with codes. And one has to ask, what are the advantages of modularization? So when we have an adapter, this adapter may map different components to other components, or in this case to another adapter. And so what we have here, compared to a situation where all components would relate to one other component or adapter, it's more unstable. So if something to A happens, a mutation, then all interactions may be affected. But if a mutation occurs here in this A, then the others will still function. So we see here a stabilization. So modularity increases stability. And if we can show that there are different mappings and codes that contribute to this, this would be a significant knowledge that is new, that we can really use codes to identify these modular structures in cells. And with regard to information transfer, we see here that we have more, let's say, channels or interaction interfaces. So you see with the arrows, you have overall, you have more arrows in such a modular structure than you would have if they are all one-to-one interactions. And this means that you have more, let's say, information transfer, if you will. And we go forward. And you have more steps in between in modular structures. And this is a phenomenon I call fading of causation. So in a code-based system, it's difficult to track cause and effect because you have always three. This is main finding. And then we can also ask if there are specific codes between bigger modules. So if each module is based on a set of codes, are there specific codes shown in the middle that mediate between such, let's say, big modules? Yeah? And we come to the point where we have to consider that in biology, cells make highly efficient use of their components. So at one point in time, the same component can mediate the interaction, same adapter can mediate interaction between different components, either at the same time or during development at different time points. An adapter mediates the interaction between other components. And this is what I call overlapping or overlaying codes. So you see this here, where we have in one situation, we have the black ones, the codes, and in another context, in a different time during development, we have, with the same components, we have other codes. And when we identify them in larger databases, we possibly then end up with different modular structures in time. That's maybe the slide, 2 slides ahead. And this is the summary that we end up with a number of codes forming a module that performs, let's say, different functions or parts of metabolic pathways. Yeah, several enzymes can be in one module, and another module has other enzymes. And this modularization allows a mixture of different functionalities. And so in metabolism, let's say you have the first processing of a nutrient molecule, and when this is transformed, maybe you have another module that is responsible for the storage, or let's say from sugars it makes fat molecules. And then another point regarding the modules is, when you look at network biology, you find these bow tie or hourglass structures. And when you say, okay, we have a module and we have this component Cx, which is in itself an adapter, you can say there are different components that are mapped ultimately through the specific adapter in the middle to other components as the output. Then you will end up with these hourglass structures that are described as they are described in the network biology, and I claim here that these hourglass structures, at the heart of the hourglass structure is an adapter molecule. And this has several implications. So with each component in a cell, C1, C4, C5, it occurs at a different quantity. So you have not one molecule or two or three, you can have different amounts of each molecule in a cell. And these are integrated by an adapter molecule. And whatever steps are in between, these different inputs, they have three qualities and three quantities. They are mapped to these different outputs that each can have different qualities or quantities. So when you look at a synapses where you have transmitter release, there can be high amounts of transmitter released or very potent transmitters at small amounts. So this is what I mean with qualities or quantities. And so this is, they are integrators. Adapters are integrators. So it's a perfect description with codes as the adapter as a kind of modulator that modulates the input to the output. So it's a bit different. When you say, okay, we have an adapter and we have a component, but here I would say more abstract, you have an input and you have an output, and the adapter mediates input to output. So you can come to this from code biology to network biology through this terminology shift. So it fits very well. And when we look at networks, so my dream would be the list of codes is much longer, so we go deep and find so many codes. And then we can go to the biological databases and annotate the databases with the codes known. And when we look then at which time point in a cell cycle or development of an organism, which codes are active, then we will see different patterns of active codes. So when we go through time, we will see that different codes are active. So in this case, we have the blue codes active at time point 1. At time point 2, we may have active these adapters or these codes. And at a third time point, we find those codes active. And from this occurrence that at the same time different codes are active, we may find hints to modular organization and have to check, do these codes belong to similar modules? Can we find modules based on the co-presence of active codes? This would be one approach to use. And what I call also code bricks are actual instances of a certain code. So let's say in the best case, the genetic code has 64 instances. Yeah? concrete mappings. I like to call this code bricks, which is also a term, this brick term is from synthetic biology and sometimes also in network biology used. So I like this code bricks as a more concrete building block. Yeah? It's not like an abstract term, but it's the concrete relation. Concrete component 1, concrete adapter, concrete component 2. This is what I call a code brick that is active through a certain time in the cell. And if we have now 3 modules, we may identify codes that mediate the interaction between modular structures. So we have on the one hand the codes per module. On the other hand, we may find the interactions between modules also mediated through codes. And this may at the same time relate to what is known as gene modules. So the question is, are these single components related or are they inherited on the same gene locus? So do we find these modules at different levels, at the gene level, at the RNA level, at the protein level? These are open questions, and which I hope to address in this follow-up paper, where I want to investigate concrete modules at all those possible levels. I have here a simple formula that can then quantify modularity. So there's a lot of literature out there about modularity and network biology, but to my knowledge, none of them has really a concrete and simple to understand measure of modularity. And the simple measure would be the number of codes per functionality overall divided by the total number of codes of a system. Yeah? So maybe a cell has 100 modules, 200 modules. This is indicated with the N. And then you have the overall number of codes in a cell. And then you get a unitless measure of the modularity of a system. This is the ultimate goal. And when you compare chemistry with a thinking of one-to-one interactions, A reacts with B and B reacts with C, when you look at such reaction networks, if you will, then it looks quite different from a code-based network. So, and this is what I call the granularity. Yeah? Each code in itself is the smallest module. So you have one adapter and 2 components. They are related. This is the minimum unit you have in my network approach. The minimum building block is a code or code brick. So it always comprises 3 concrete molecules in a cell. And this leads to this distinction between the granularity and, we are already at pickup slides. So this was just a visual approach. As I said, for all who want to know more about this, there is the paper of the same name that describes this in more detail. I want to just give a visual approach that you have an easy account to see what this means for network biology and what one could do when one really categorizes all components of a cell and looks, what is this component, what is this molecule, is it part of a code, is it not part of a code, and then make such kind of analysis. And I want to show further slides that are not related to the modularity, but to other topics that emerge out of this code view of life, if we have enough time. So the 3 A's of the mind, artifacts, archetypes, and otherisms. I really was inspired by Joao's paper, that these archetypes are patterns of behavior that are inherited, and how this relates to codes. So in biology, you have different what calls atavisms. So atavisms are, let's say, remnants of the past. They are many anatomical structures of a historic species that occurs in a later species. But you can also say that there are behavioral mechanisms that appear from the past in the present. And when you look at similar behaviors or types of behaviors that are elicited by visual recognition of a puppy, baby, child, adolescent, or adult. So let's say a lion can either protect a puppy if it's his own, or he can kill a puppy if it's not from him. So it's a way to relate in a code-based manner self versus non-self, or mating behavior, male versus female. So these are all mappings that fit this code model very well. And as you have mouse here, there's it. There's it. So you have this, what we call misbehavior. In biology, it's called this displacement behavior. But somehow these all are, let's say, archetypes, behaviorally determined outputs that are mapped by the human brain as an adapter, for example, to a certain input. So they really fulfill the criterion of code. And it's a bridge, let's say, or at the highest end of codes, at the highest level of hierarchy are the cognitive codes that I wanted to address here in this slide. But as Marcello mentions in his publications, there are the molecular codes, and then he jumps to the cognitive codes where he says the brain is an adapter. But the question is there a continuity from the very bottom to the highest level of codes? And I have a somewhat different approach than Marcello, especially regarding meaning. And currently I'm trying to resolve to fill the gaps from brain to molecules. So if there is a way to describe how molecules become codes and how codes ultimately become behavior. And it appears a bit complicated, but it's in effect it's quite easy. So you have at the pre-code level, you have bond makers that create polymers, and you have copy makers that make copies. And ultimately then you have two worlds and the code maker that makes the code. And what I call code users, they ultimately produce artifacts. So an artifact is the first occurrence of a non-natural molecule. And with this, we have the real novelty in the abiotic world and the transition to the biotic world with the artifact. And then the question will be if we can have a number of artifacts and meaning makers that make meaning out of those artifacts, because I do not see meaning in a code in itself. It needs another instance above, a reference frame that makes meaning out of codes or products that are produced by code users. And then from those meanings, we end up with interpreters. And from interpreters, we end up with sense makers. And we have a transition from natural laws to rules. And then it comes into play the concept of agency. And we see that we have here also different directionalities related to those underlying processes ongoing. This is what I'm currently working on. And just to give you an outlook of what it means. So when we have a code user that produces artifacts, we can have a meaning maker that would make, as the name says, a meaning out of two artifacts. And therefore, at least it needs two codes to make meaning, because two codes that are involved in an artifact production make those artifacts. And from those artifacts, a new meaning can be derived. In general, there is a thing, this is, I guess, not addressed by Marcello. There are artifacts that are part of codes, but there are non-artifacts, like in the signaling codes, calcium. Calcium is a biological molecule. It's abiotic, it's a chemical, but it can be part of a code. So you have to distinguish. There may be codes that rely on a mapping between two artifacts, a mapping between an artifact and a non-artifact, and maybe a mapping between two non-artifacts. So there's really a new dimension of what is necessary to consider how meaning can be derived, or the possible pathways to meaning from codes and artifacts in this case, because artifacts are the first non-biological molecules here. So this was just an outlook, and I want to point towards the latest paper I have out, which shows not only how modularity, but how hierarchy relates to codes. And I have this free link, which is only accessible until 9th of July. And you can download this from Elsevier for free via this link. So otherwise this will expire. It's then not free anymore. And with this, I'm finished and happy to answer some questions. Thank you.
We now have some time for questions, as the next presentation Adam wasn't able to attend. I would like to know if anyone has questions for Roger.
Well, thank you for your nice talk. You showed a lot of entities, which seems to me to be a graph. You go next some slides to see a graph-like representation of the codes. And I was wondering, is there any electronic representation already available where you can retrieve information from existing codes?
No, from existing codes, not. This is the problem. So do you mean this kind of what?
Many codes, right? I mean, the list is how many hundred of codes?
Well, it's 250 now, about.
I'm asking the question because we could retrieve information if we had such a representation, and we could do also other things with the codes. Do some more analysis if we had an electronic representation structure, data structure.
I mean, the main problem is to really retrieve information, it would need much more of the code. So the problem with the code list is, it's a collection of all codes. Yeah, it's from neuronal codes. Yeah, many cognitive codes, recognition codes, pattern recognition codes, how the brain works, to molecular codes, sulfation codes, genetic code, epigenetic codes. So if you want a meaningful analysis, you have to go to any model organism check if any of the 250 codes is there. And then the problem is you will end up for a bacterium, all of the neuronal codes, they are not there. And so you have very few codes left. So I fear that for this kind of analysis, we need much more codes to be added to make a real comparison. The other thing would be to have a minimal organism and to check what are the coded relations in those very minimal organisms.
Yeah. If I may add, the question was not related to a specific organism, but to the structure of the code. So for instance, the genetic code, 64 codons mapped to the amino acids. And I'm not familiar with the other kind of codes that are there, but I'm pretty sure there is a similar structure and you can just try to adapt them.
I see what you mean now, yeah. This is a point I also want to address in the near future, is a deeper annotation of this list. Because what we have in a list is the genetic code and, for example, the phosphorylation code. We have seen in the quorum sensing the phosphorylation. Phosphorylation is one of the most important codes in organisms. Yeah, it's everywhere. So, and I propose for the genetic code, we have the best case, we have 64 instances. Yeah, but for the phosphorylation code, we have much more instances. Yeah. And this type of structure, yeah, this can be reflected already in the database now. If one goes to it, look, what is the phosphorylation code? How many instances are there? And then we can make a more general comparison between all those codes. Yeah. This is what we can do. Yeah.
Maybe clustering of the codes to find similarities, etc.
Yeah.
Yeah, so we discussed already about this, but I think it could be a very general purpose of our society to find a code classification. For example, like Lamarck began in the 18th century to classify organisms. And I don't think it doesn't matter where you can find this code. It's maybe a very specific question, but just kinds of codes.
This is what I would call code cladistics, yeah, something like this, and where you can evolutionary follow when did all those codes appear. Yeah, I mean, if I had the manpower, PhD students, master students, you can put them. Say, you have the codes, you take the first 20, next 20, next 20, go, what is the code? What is the origin? When did this genetically evolve? And just do this type of evolutionary analysis and find when did they develop, in which context. It's not so complicated.
No, it is not what I meant, and it is not a work for students. You need some more oversight to do something like this. I mean, the classification from the top. And it is not related to organisms or to any details. I think what we lack is this structure. We should find some structure in this. No? And I mean, this society is actually ideal for this because we have specialists in different areas. For example, I cannot say what cultural codes are. I cannot distinguish between. But we have also people in our society who can.
Yeah.
I have just two quick questions, please. The first one relates to the mathematical formula you presented. I wonder what was the unit for the equation?
It has no unit.
It has no unit.
And this is the big advantage. Also for my complexity, I have another paper in this which you can download. I propose this formula about complexity. Yeah? And the big advantage is you have no unit. Yeah? A code has no unit, and the codes per system under investigation has no unit, and this allows you to compare completely different systems. You can compare a mitochondrion with a human. Yeah?
Yeah, but do you think we can call it mathematical if it has no unit? Do you think we can? I mean, call it math if we can divide a quantity by another quantity. I mean, is it really a relaxed term to say?
Maybe it's not. Maybe it's not a formula. You can call it a ratio, or
Okay.
yeah.
And my second question was a quick one. The directionality you mentioned, it seems that meaning is coming before interpretation. And
Yeah.
yes. And do you think we can get meaning without interpretation?
I think we can have meaning, objective meaning, without interpretation. Interpretation, it would be need too much time to discuss this now. But yes, I think there is meaning objectively without interpretation. But interpretation is based on this objective meaning. That objective meaning is based on artifacts, yeah, basically, yeah. And you have this continuation from bottom to top. This is what I'm currently working on, yeah.
How robust are these categories that you're using for defining code? For instance, let's imagine it's a language. Would you have that the Italian is the same as German or different? You are categorize this as a language or specific language? Or because it is, I mean, you know, in Italy we have 22 different versions of cappuccino. You can order, we did it for fun. We can order cappuccino in 22 different ways. I believe in Germany, maybe you have 2 or 3. Cool. But what I'm saying is that then would be the same language, would be the same code. How you are defining code? Because code is
it's a code. It's the same. I'm not a linguist, but from my perspective, it is the same category of code. It's like the genetic code, but this is an instance, yeah, a concrete instance. They may say it's a dialect or whatever, but for me it is an instance, yeah. Overall language, what instance? German, Italian, any other.
You are mapping sets that are different. The same word is mapping different sets, so they are different codes. So I mean, I'm not sure, but I think unless you are addressing the operationalization of the meaning at the moment you are decoding the code, talking of code in general is to me.
And I would say it's a language code. And what you have next, cladistics, then is the cappuccino code. And the cappuccino code in Italian has 20 instances, and in the German it has 3 instances. Yeah, like a genetic code with 64 instances, or you can have a simple genetic code with 40 instances.
Yeah, but it's how you handle that in this type of analysis, because you will have different things.
No, you would not have differences.
Noise and precision measurement.
However, that's okay. Not just to clarify.
You know, I would analyze a system. I go to a German guy and think, tell me all about cappuccino. And then I have the concrete instances of cappuccino use. This is how I analyze it. Then I go to Marcello and ask him about cappuccino. And hey, I count 23 instances of this.
Mm-hmm. So you do this for all the language in the world, all the C they are there, including enzymes, whatever.
Yeah.
I mean, I think that at a certain point is not handled.
Yeah, this is the thing. There are hundreds of thousands of instances of codes, and people still here maybe not realize what this really means. And this would be the next step. If there's any informatician who can train a system to automatically identify from all those big databases, yeah, where 20,005 proteins, RNAs, map those. Yeah.
It seems to me that it will be much help to work with relational databases.
Yeah.
In analyzing so many data, similar, dissimilar, and to be efficient in that. So you will probably need some relational data management.
This is the problem. Yeah, I have the concept, but I have not the concrete skills to extract all those data in a way I want it from those databases. This is a problem. Yeah.
That was exactly why we wanted to do a relational model, because when person who made FASTA, and which is a standard bioinformatic format, was doing that in early 2000, and They started in 2010. They were relational databases for the data of DNA. But the problem which arises is the analysis. The code itself was not tuned to terminology. So we have a huge relational databases of sequences, but we needed an annotation of elements, A, C, G, U, to calculate with polymorphisms and some specific aspects. So probably it is a very similar problem.
The point is you need a few guys who are doing this manually with our intelligence, with our real intelligence, and hopefully a clever guy who can train the computer to then extract the other things automatically.
Sorry, we have time for 1-2 questions more, please.
Yeah, I will.
Yeah, sure.
May I just comment to Modular? To the mic?
Can you hear me? It's for no. Okay, I just like to add that relational databases are a specific form of a database which stores data as a table. And for me, such a structure of codes has another structure, which is probably where another kind of database or data structure is more suitable. So I would be open for any kind of storage format, but I think the point is we need to describe the data, give them a structure, and then store it to be able to retrieve it. Yeah.
Just a comment. Here in the CCG, we have a set of projects where we do not use relational databases, we use graph-based databases. So the mapping is direct. We can map directly this network into our graph-based databases. And we use that for studying meaning and semantics of information. So we do work in ontology engineering. So if you need any kind of help, we can try to map your networks, to extract meaning, to construct an ontology, and to have some kind of rationale about the compositionality of modules. You use the word overlapping and
Overlaying.
Yeah, for us that is called compositionality. So we can study the composition of modules using our graph-based databases for constructing an information ontology to understand what you are trying to study in the biological world.
Let's do it. Show how graphic design can be, because you have patterns.
Yeah.
You have within tables, you have patterns which you can calculate graphically. So that's precisely.
We have also some functions present visual information about the meaning that we extract from the graph-based database. Yeah.
You're right.
Yeah, I never went. I was alive for 5 spots just to register. No, you made me think about the attempts that started 10 years ago about modeling the regulation of transcriptional networks. And it went both 2 ways, like John Tyson and Belanovic with differential equations and so on. And Uri Alon, for instance, which took patterns of connectivity and signal transduction pathways, so phosphorylation and so on, and from there established codes, really, linked molecular connectivity to cell behavior. So I think some of the work is already done, not manually, because he's an engineer. It's a Israeli guy. He's a molecular biologist and an engineer. So maybe it might be useful to have a look at.
Please send it to me then.
Uri Alon.
You wrote a book about regulatory motifs.
Yeah. The motif.
Yeah, but Uri Alon, you can just look up.
I have the book, yeah. This blue book with the orange. Yeah. Yeah. Okay.
Can I just, this time, you've got this formula. You can count the number of codes, right?
Yeah.
But what do you do with functionality? How do you count functionality? Because functionality is in the eye of the observer.
No, not so much, because you can count the enzymes that are involved in carbon metabolism. Let's say you have the lac operon, yeah? You have glucose or you have lactose. You can clearly identify all enzymes and all steps that are involved in the metabolization of lactose and of glucose. Yeah, it's clear. It's not arbitrary. No, not of an observer. You can say, and we have them, and you find this functionality, how many codes are involved in all those steps metabolizing glucose.
Okay, but then we have to talk about what you regard as a code, because they are all deterministically linked. Enzyme reactions, there's no code in metabolism. There's no, the connections are all the whole network of metabolism, all the connections are deterministic. So then we have to figure out what you call a code. It's the same with the metabolic
That's
code.
questionable. That's questionable. Yeah. I mean, don't make that statement. No COVID. Wait a minute.
We're going to differ about this.
Yeah, okay.
Always.
Just want to address two points of this.
Flat network.
One thing is about what Mario is talking about. Yeah, I think that the things that are mentioned, it's about how to study code. Because from my point of view, you are looking for a top to bottom view of the codes, because you want to categorize and then want to see what are the specific codes linked to those modules and all the stuff. And from the point of view of Mario, he wants to see that, okay, we have hundreds of codes, and then how they come together to become the major code that you want to build. So I think you are looking for the same, but only in a different directions of the study. Yeah. And the other stuff is related to what Ricardo mentioned. I think it's something important to start building this ontology of the codes. That is something you are indirectly begin to work on. And I think that if we can categorize and build this on ontology of the codes as something that Elena tried to also mention too, we will have a better understanding of the general mechanisms that Marcello already stated, and then continue to the evolution. And we can also integrate the biological properties that we're having. We have modularity, hierarchy, and all that stuff. So I think it's an ongoing project that we need to continue, because it's the second stage of the code biology. We already settled, but we need to build all these general ways to study codes, I think.
Yeah.
And we need different tools.
Yeah, we have a lot of them. We already have this graph-based analysis, network biology. We already have deep learning. So we have a lot of tools to work on.
I think that one thing that came from this discussion is that maybe we are talking about different levels of complexity, and the modules that organize the codes might be on different levels, and maybe those adapters that you suggested will be the link between the model of the genetic code to the model of the metabolic engineering, metabolic pathways, although you might not consider it code. But now we can see these, we can categorize maybe by the modules and link these modules together to build a complex system that could describe the organisms. And I think that what I got from all this discussion, that, and basing on your principles, that maybe these models are on different levels, then we can add a complexity turn to your functions, and maybe that could be the link between them. I don't know if you agree with that.
I agree, yeah. So more of this is in this paper with this download link. There's a discussion of the complexity. I wanted to focus on modularity today, but in general with categorization, I propose a model with two very fundamental type of codes: regulatory codes and structural codes. So structural codes relate to fundamental processes, yeah, and the fundamental processes, they can be adapted to different circumstances, and therefore it needs the regulatory codes. So in those regulatory codes, they would relate to what you call this more at different levels of integration. Yeah? You can find them there. Yeah.
Just add a few additional hints. I've talked with Robert yesterday.
Yeah. Yeah.
When we study modules as a way of measuring networks, or as a way of understanding the way complex systems organize themselves, we can use or we can study two different properties: cohesion and coupling. Cohesion for us is very important because we can understand how internally all the minor elements that compose the model are organized for giving them the behavior that we can observe from the outside, which may be good for understanding how codes can be composable for forming a model. When we talk about cohesion, for us it is very important because they explain, or they are mechanisms for explaining the way modules can be composable, which means how they can interact and form a more complex organism. Which means that if we study your network based on the construction of ontologies, we may clarify the properties of cohesion and coupling in these biological systems. Just another hint and another suggestion.
I think you could start working together on the database. You know, keep that in mind
Yeah.
when you're thinking along with the database.
Thank you, Robert. I think that's all for the discussion, right?
Okay, thank you.
Thank you.